So I will start again. Good morning to all of you. In the last class, we have studied about the nerve impulses, how the nerve gets uh, or helps in the transmission of the impulses from one point to another point. And for this, we have discussed about few important things like resting potential, action potential, what is depolarization, how the depolarization is brought about, and all these things we have discussed. And now today we will continue with what we have studied in the previous class. And uh, here just I would like to draw your attention for one more terminology, that the refractory period. So there are two types of refractory period. One is known as absolute refractory period, and whereas the other one is relative refractory period. So this absolute refractory period is a period of about 1 millisecond, during which the impulses can be propagated however intense. No impulses could be propagated however intense the stimulus is. So for the uh, impulses to propagate, there should be an impulse, and this impulse is responsible for changing the status from resting potential to action potential. But this stimulus, or when there is a stimulus, the impulse should be propagated. But there is a refractory period of 1 millisecond. Within this, however intense our stimulus may be, the impulses will not start propagating. That is known as absolute refractory period. Another one is the relative refractory period. This is slightly takes more time, 5 milliseconds, and during which the new impulse can only be generated if the stimulus is more intense than the normal threshold. There is a normal threshold below which no impulses can be generated. But this lasts for 5 milliseconds, and already one impulse has completed. And if you want the second impulse to be generated uh, with more intense stimulus, the stimulus should be more intense, but even then it takes 5 milliseconds from one impulse to another impulse. First impulse, okay. For the second impulse to start to generate, it must be uh, more intense, and it should be more intense than the normal threshold period. But in spite of that, it will take another 5 more milliseconds. So here is a graph where you can see uh, this is the timing in milliseconds. So this is the neuron excitability, and here you can see this is the stimulus and the time. And normal time is about 2 millisecond. Normal these things when there is a stimulus, then up to 1 millisecond no impulse can be generated. That is known as absolute refractory period. But the next one is, it should be the intensity of the stimulus must be much more than the earlier one. Then the second will start, and that is known as relative refractory period. And once the impulses start conducting, immediately the portion of the neuron after conducting the impulse comes to the resting potential. This is how you can expect. And so now the question is, what the importance, what the relevance of this refractory period? So this refractory period makes the flow of the impulses only in one direction, because the reason beyond the impulse you cannot depolarize it, because unless there's a depolarization of the neuron, no impulses can be conducted. So what the refractory period makes, the ensures that the impulses can flow unidirectionally, as the reason just behind the impulse you cannot be degenerated for at least for this refractory period. And then It limits the frequency at which successive intervals can pass along the axon. For the information to travel or the impulses to travel, there may be so many stimulus, but simultaneously you cannot send all those stimulus through one single neuron. So there must be some gap, and this refractory period ensures that gap of 0.1 milliseconds to 5 milliseconds. This limits the frequency. Even if you within that, if you have more number of stimulus also, they are not conducted unless the first once the first impulse is traveled, there must be a refractory period, and then the neuron becomes ready for the next impulses. So that's why this refractory period ensures two things. One is the unidirectional flow of the impulses, and the second one is the it limits the frequency because it will prevent the overlapping of the impulses one after the other with a gap of 1 milliseconds or 5 milliseconds. So this is the use of this refractory period. Now this photograph, you can see that how the process of neural transmission happens. So the first one, which is marked as A, this is a polarized membrane where you can say it is in the resting potential. See, outside positive, inside it is negative. When the impulses hit one point uh, in the form of stimulus, for when the stimulus hits this one, impulses are to be generated. For this matter, what happens? The positively charged sodium ion flow in, and as a result, there is a change in the positive and negative charges. Outside become negative and inside become positive. This you can say that the initiation of the action potential. And this is now you can also show that the membrane is polarized, depolarized. Earlier in the resting potential it was polarized, but when the stimulus has hit, the sodium ions flow in, changing the positive and negative charges. Outside during that portion become negative charge, inside it becomes positive charge, and that is what you call the initiation of the so-called action potential, and that is achieved by the depolarization of the membrane. Then once then the stimulus is in the form of the impulses, and it is moved in this direction, the direction in which the arrow is shown. Once the impulses move to the next stage, already depolarized portion of the membrane becomes normal. This portion becomes uh, polarized, but whereas the next portion becomes depolarized. Here, the sodium ions move inside, but in the previous area, the sodium ions are pumped out. So in this one, the one which is shown with the blue arrow, that becomes depolarized. That becomes polarized, whereas the next portion, which is the shown with the black arrow, that becomes depolarized. So as a result, what happens? The uh, impulses move forward. See, then again this get repolarized by pumping the sodium ions outside, and the next portion becomes depolarized. So like this, you can expect the process of neural transmission, that the impulses travel all along the neuron. So here also you can see here in this one here the action potential has generated because of the change in the positive and negativity, and then in the next one it moves to the next stage, but the previous stages portions become polarized. Only the portion where the action potential is shown, that gets depolarized, and thus they carry the impulses. In the very beginning or in the last class, I might have told you there are two types of nerves. One is myelinated and another one is non-myelinated. They are also called as medullated and non-medullated. In a myelinated, axon portion of the neuron is covered by a protein by name myelin sheath. Why this myelin sheath is important? In highly advanced organisms, or not highly advanced, most of the advanced organisms, the neurons are myelinated. In a neuron which is myelinated, electrical signals travel as a continuous wave. But in the non-myelinated axon, there are insulators. Insulators are the one which prevent that the conduction is stopped or it is slowed down. But in unmyelinated axon, the electrical signal travel as a continuous wave. But in myelinated axon, unmyelinated axon, this prevent the flow of the impulses. But in myelinated one, what happens is the signals, instead of traveling uniformly, they have to hop. Because if you can go back to the earlier portion, just I will go back to that slide, because unless you see that slide, it will be, yeah, this is, see here, this is a myelinated neuron. This is the myelin coat, and the myelin coat is not continuous. See, there are many interruptions. But in unmyelinated one, there is no myelin and there is no interruption or anything. So, but the signal flows as a continuous wave, but it is affected by a lot of insulators. Here also, the myelin sheet is not continuous, so that's why at the end of each myelin gap, there is insulators. So this causes the signal to hop from one myelin sheet to another myelin sheet. So these parts in between these insulators are called nodes of Ranvier. Nodes of Ranvier are nothing but the insulators which are found in between the myelin sheaths. So these are insulators and they prevent the uniform flow of the information, and that is known as nodes of Ranvier. And the action potential will hop from one node to another node. See here, this is unmyelinated one. The earlier one is myelinated one. The information has to hop from here to here, here to here. Because this node of Ranvier, they are insulators, so they cannot flow uniformly, but they have to hop. But in case of the non-myelinated one, where you can see in the second photograph, the there is no node of Ranvier, so the impulses have to travel all along the axon uniformly. But here they can jump here from one myelinated sheet to another myelinated sheet like this. See, so I will give you a small video clip where you can see. See, the first one. Can you see the video? Hello.
Yes, sir.
Okay, in the first one, which is non-myelinated, see how slow the information flows, because they have to flow all along. But whereas in the second one, which is myelinated, you can see the information jumps from one node of Ranvier to the next one. So the conduction of the impulses is much faster in the myelinated one and much slower in the non-myelinated one. I hope this uh, clipping will give you the how the information or impulses get transmitted. So these electrical potentials travel faster in the myelinated axon compared to non-myelinated or unmyelinated one, because myelin is an electrical insulator which does not allow the conduct of electricity. In our body, we have both myelinated as well as unmyelinated axon, but myelinated axons are much more. These myelinated axons are created by the Scavon cells, and the oligodendrocytes. These are the cells which give rise to this myelinated axon. So as a result, in the myelinated axon, the electrical potential travels faster than on unmyelinated ones. So now we have seen that the travel of the impulse within a neuron. But we know in a cell there are innumerable trillions of such neurons. They form the nervous system. So as a result, the information should be carried from one neuron to another neuron. So here I have shown you two neurons. This is the one neuron which receives the information. Then the nerve impulses are conducted along the axon and it will reach the next neuron. Especially the dendrites of this next neuron will receive the information that is coming from the previous axon. So this is what we call the transmission between the cells, and the transmission between the two neurons or between two nerve cells we call it as synaptic transmission. See, uh, here this is one neuron at the terminal end of the axon is also branched and they are having button shaped structure. Can you make out in the end, in the axon where I am moving the cursor, you can see that the every neuron at the end, especially in the axon part of the neuron, is branched and it is forming a cup or a shaped structure. And the process here, the transmission of the neural impulse between the cells, that is, to be more precise, from one axon to the next dendrite of the next neuron, the axon of the previous neuron to the dendrite of the next neuron. This movement we call it as synaptic transmission. This is known as synaptic transmission because the axon of the previous neuron and the dendrites of the next neuron, they form a junction, and this junction what we call synapse, and the transmission across the synapse is known as synaptic transmission. For your information, the axon of the one neuron, earlier neuron, and the dendrites of the next neuron, they are not physically touching each other. They are not physically touching, though they are in very close contact, but they do not physically touch each other. Instead of that, the axon of the previous neuron and the dendrite of the next neuron, there is a small gap, and this gap is known as synapse actually. See here the axon, then axons branch and form the buttons, what you call synaptic buttons. Within synaptic buttons, you can see lot of mitochondria and the vesicles, and it is externally covered by a presynaptic membrane, and then there forms a gap. That is what is called as synaptic space. Then here comes the next neuron, the dendrite of the next neuron. This also externally covered by postsynaptic membrane and dendrite. This gap between, this is the first neuron, this is the first neuron, this is the second neuron. This is the dendritic part of the first neuron. This is the terminal end of the axon of the first neuron, and the gap which you can see here, it is known as synaptic gap or synaptic space. Now, for the conduction of the impulses, the information coming from this neuron has to reach the next neuron, bridging this gap. What is this gap? That is known as synaptic gap or synaptic space. So, this transmission of the nerve impulse from this axon of this neuron to the dendrite of the next neuron, we call it as synaptic transmission, because it has to pass through the, cross through the, or bridge the synapse. So what is this synapse? It's a very, very interesting thing. Thus, our central nervous system contains more than 100 billion neurons. Our central nervous system, that's the brain and the nerve, spinal cord together, they contain more than 100 billion neurons. So the incoming signal enters the neuron through the synapse, and these synapses are located mostly on the neural dendrites, but they are also on the cell body of the previous one. The different types of neurons, there may be only few hundreds or 2 lakh such synaptic connections from input fibers. Though there are 100 billion neurons, you don't see 100 billion synapses, but the number of synapses may be a few hundred, 2 lakhs. And conversely, the output signal travel by the way of single axon leaving the neuron. Now, what is synapse? Here, just you can again see the exact. These are all the synaptic terminals and how the dendrites and axons they unite. The next picture will junction where axon and some other portion of the cell terminates the dendrite. That is what is called as synapse. As I have told you, that the junction between the terminal axon of one neuron and the dendrite of the next neuron. And this term was introduced by a British neurophysiologist by name Charles Sherrington. Charles Sherrington, who got the Nobel Prize for this, to explain the synaptic transmission. Now, let's see what actually happens in the synapse. Information, all of you know, gets transmitted in the central nervous system, uh, mainly in the form of action potentials or nerve impulse, which pass from one neuron to another neuron. And naturally, each impulse from on its way has to pass from one neuron to another. And this information sometimes may be blocked in the transmission from one neuron to another neuron, or sometimes it can be changed from a single impulse to repetitive impulse. This is also possible. It may be a single impulse, but it may be a single stimulus and a single impulse when it has started, but it may become repetitive impulses. So that's why this synaptic transmission is a very complex process which helps in grading and adjustment of the neural activity, which is very, very essential for the normal neural function. So this synaptic transmission is very, very complex process because it grade and adjust the neural activity, thereby influence on the impulses, thus ensures normal functioning of the neuron. See, this is the cell body or cyton. This is the axon of this one. See, this is here. How they communicate. But there is a gap. It is not actually touching. The terminal buttons, synaptic buttons are not touching the axon of the next neuron. But there is a gap. Very small gap. So we have different types of synapses. One is exodendritic synapse. Synapse between the axon of one neuron and the dendrite of the another. We call it as exodendritic. If this junction is formed in between the axon of one neuron and the dendrite of the other, we call it as exodendritic. The other one is exosomatic. It is a synapse between the axon of one neuron and the cyton of the other, soma or cyton. Axon of one neuron and the cyton of the head portion of the another neuron. Apart from these are two basic types, important one. But we have three more. One is exo-axonic, where the synapse is between axons of one neuron to axon of another neuron. Another one is dendrodendritic, where the dendrite of one neuron communicates with the dendrite of the other neuron. And the third one is dendrosomatic. Dendrite of the first neuron communicates with the cyton of the next neuron. So this is known as dendrosomatic. So totally there are five types, but exodendritic and exosomatic are the major ones. See here, exodendritic. So this is the cyton, and this is the dendrites, and this is the axon. This A, this is the one what we call exodendritic. This is exosomatic. See, this is the axon of neuron which communicate directly with the soma or head or cyton of the neuron. Then exoaxonic. See, this is the axon of this and this is the axon of this, and both of them join and communicate. That is known as exoaxonic. So there are different types of synapses. Then there are also different types of synapses which are classified based on their functional capabilities. In that, the first one is chemical synapses. Almost all synapses that is used for the signal transmission among the human beings are chemical synapses, because here some chemicals are used to bridge the gap between the two neurons. That is, first neuron secretes the chemical substance, and that chemical substance is known as neurotransmitter. Actually, the signal which has come from gets transformed into this neurotransmitter, the chemical, and this chemical reach the next neuron. So here, to bridge the physical gap between the adjacent neurons, a chemical is secreted, and such new synapses are known as chemical synapses. And these chemicals which are secreted, they are known as chemoneurotransmitters. And these neurotransmitters, that the chemicals released from the first neuron, binds with the receptor site of the next neuron and excite it or inhibit it or modify their sensitivity, and thereby they carry the impulse or they transmit the impulse to the next neuron. Second one is the electrical synapses. They are less common compared to chemical synapses. These electrical synapses are less common. The membrane of pre and postsynaptic neuron, they come together. They are close enough. But still then there's a gap junction. So here what happens, instead of chemicals, electrical signals are released from the first neuron, and that bridge the gap and reach the next neuron. And especially they are less common compared to chemical synapses, and um, are important in the CNS also, but most of the CNS is chemical synapses. Central nervous system is having chemical synapses, but in some, especially for arousal from sleep, mental attention, emotions, and memory, there also you have to conduct the impulses, and these impulses are generally in the form of electrical signals. Even iron, water, homeostasis, all in these things where the information is passed in the synapse in the form of electrical signals. Now, talk whatever may be the type of synapse, whether it's a chemical or electrical. Let's try to understand the anatomy of the synapse. See, this is one synapse. This is the axon terminal of the first neuron. I have already told you there is a mitochondria and ATP because it's an energy intensive process, and so that's why they need energy. And here in the synaptic clefts or bulb, you can see lot of vesicles, and these vesicles they contain neurotransmitters or the chemicals. Then comes a gap. This is known as synaptic cleft or synaptic gap, and this is the presynaptic membrane, and this is the next neuron, and this is what you call the postsynaptic membrane. It is the dendritic section of the next neuron. This is the axon section of the first neuron. This is the dendritic section of the next neuron. See, this is how it appears. That's a gap. This is the axon terminal of first neuron and the dendrite terminal of the next neuron. So this, what is the size of this gap? This gap, what you call synaptic cleft or synaptic gap, is very small gap, about 200 to 300 angstrom units. This is the space between the axon terminal and sarcolemma. Axon terminal and sarcolemma of the next dendrite. Sarcolemma is nothing but the plasma membrane itself is known as sarcolemma or sarcoplasm. So here the width is about 200 to 300 angstrom unit. This gap, the one which you can see here, the gap is about 200 to 300 angstrom units. Now, at the presynaptic terminal, there is a synaptic knob, and this presynaptic knob cover about 40% of the cyton and 70% of dendritic membranes. These presynaptic knobs are formed by the contribution of both dendritic membrane as well as the cytonic membrane. 40% from cytonic membrane and 70% from dendritic membrane. See here, one more photograph, the chemical activity which you can see here. It's the information gets transmitted by the chemical what is known as neurotransmitters in different steps. I will come to the steps. So at the same time, here you can see on the presynaptic membrane, you can see lot of vesicles which are filled with what you call the neurotransmitters. And on the other hand, on the next membrane, that is the postsynaptic membrane of the next dendron, there are many receptor sites here. There are many receptor sites which are ready to receive the chemical that has been released by the presynaptic membrane of the first neuron. So, large number of the postsynaptic neuron contains large number of receptor proteins. Now, what is the fate of these neurotransmitters? Basically, the neurotransmitters are bound to postsynaptic membrane. Probably here you have seen. Neurotransmitters are found in the presynaptic membrane. So the transmitter found in the presynaptic membrane, and they have to bound with postsynaptic neuron. And as a result, they produce a continuous postsynaptic effect. At the same time, they block the reception of additional messages also. Only this message which is carried by the neurotransmitter is to be conducted. It will block the reception for additional message. After the neurotransmitter released at the synapse, then it is removed by diffusion out of the synaptic cleft into the surrounding fluid, and they cross the surrounding fluid, and acetylcholine esterase, an enzyme destruction will happen, and the vesicles will dissolve active, and the chemicals will reach the next one, and active transport, and again these neurotransmitters are brought back to the presynaptic terminal itself by again the enzymes, for example epinephrine. So here, before going to this, I will go to the earlier photograph where I will be able to explain the transmission step by step. So here, in the first instance, in this postsynaptic knob, the presynaptic knob, vesicles and peptide neurotransmitters are synthesized, and these neurotransmitters are bounded by the vesicle. Now they are ready for the transport. From here, they come to the presynaptic membrane, and now the vesicles are storage small neurotransmitters. They store small amount of neurotransmitter. Now the action potential causes the calcium ions to enter and trigger the release of this neurotransmitter. Here there is an action potential is generated, and it causes the calcium ions to enter this inside and trigger the release of these neurotransmitters. And these neurotransmitters are released, and then the neurotransmitters molecule cross the synapses here, which are bounded by the vesicle. The vesicle is opened out, and the neurotransmitters are released into the cleft, and the neurotransmitters or the chemicals have the capacity to cross this synaptic cleft. And once they cross the synaptic cleft and touch the postsynaptic membrane, neurotransmitters are attached to the receptors. Here there are receptors. Can you make out here? The receptors. And these neurotransmitters get attached to the receptors, and it starts postsynaptic activity. And these chemicals contain the information which has come in the form of come along with the axon, and now the information in the form of acetylcholine or chemicals has entered into the postsynaptic membrane. Then what happens? The chemicals, they touch the receptors and give the information. Once the chemicals, the neurotransmitters, give the information, reuptake of the molecules takes place here. Again, this neurotransmitter go back to the presynaptic membrane, and neurotransmitter molecules broken up by the enzyme, especially monoamine oxidase, and as a result, the these neurotransmitters are broken. So neurotransmitter, first they are bounded by the vesicle, cross this presynaptic membrane, release their content in the cleft. Clefts reach the receptors on the postsynaptic membrane, pass the information. Once they pass the information, reuptake of these uh, neurotransmitters takes place by the previous neuron through the presynaptic membrane, and once they cross the presynaptic membrane and reach the bulb, that is the axon bulb, there these neurotransmitters molecules are broken up by monoamine oxidase. This is what happens here. So this, here, this is the entire philosophy or physiology that I have told you. But this is not as simple as I have told you. There are lot of presynaptic physiology and mechanisms are derived here. First, the synthesis of neurotransmitter. See, in the axon terminal, the synthesis of the transmitter takes place, and it is alpha methyl para-tyrosine. See, then axonal transport. Then these neurotransmitters are to be transported from the region of their production to the actually to the synaptic bulb. That is the uh, axonal transport. Then they should be able to conduction of the action potential. How? By moving the calcium ions inside and potassium ions outside. They uh, these chemicals are released and they attach to the presynaptic membrane, and then modulation of the transmitter release them into the synaptic cleft, and these chemicals attach to the receptors on the postsynaptic membrane. See here in the postsynaptic membrane, the first one is inactivation of the transmitter. Once the transmitter attaches with the receptor, that gets deactivated, and it is again re-uptaken. It is sucked back by the presynaptic membrane. Then now the information with these receptors. See, there is the one which are meant to receive the chemical messenger, that the neurotransmitters. Once the neurotransmitters attach, then what happens? There is an alteration of the number of postsynaptic receptors. There is an alteration, change. Some will they are altered, and then some of the receptors are also blocked. The one which is shown with the red color. But whereas some receptors are activated, and they will recognize this chemical and the information that is carried by the chemical, and that causes the activation of secondary messenger, what is known as the lithium, and that converts the chemical signal again into electrical impulses. So here totally you can expect the postsynaptic neurons and the neural soma and leakage. Of course this is not important for you. Yeah. In order to achieve this, one has to make the synaptic excitation on the postsynaptic membrane. So this is known and make the excitatory postsynaptic potential, EPSPS. Only when this excitatory neurotransmitter bind to the receptor on the postsynaptic membrane, there will be partial depolarization. I have told you the neurotransmitters or chemicals, they carry the information and they should bind on the postsynaptic membrane, and the neurotransmitters which bind with the postsynaptic membrane, that is on the receptors of the postsynaptic membrane, we call it as excitatory neurotransmitter, these chemicals, and once they bind, they cause the depolarization in the postsynaptic cell. How the depolarization is caused? Because of the sodium influx. Sodium from outside the membrane rushes inside, and this makes the action potential to generate by depolarization, and this potential rises enough to the threshold level, and the action potential will develop and excite the neuron, and thus it carries the information to the next neuron. So here this summation of the impulse transmission on the membrane is possible provided the membrane potential has to increase from minus 65 millivolts to minus 45 millivolts. There should be an electrical gradient. Earlier, the membrane potential was about minus 65. It should change into minus 45 millivolt. Then only the summation of this will start. So there's a difference of plus 20 millivolts, which reaches the membrane almost to the firing level, and the this will cause action potential develops, action hilot, and discharge of single presynaptic terminal can never increase the neural potential. But the discharge from not from a single presynaptic terminal, there are number of presynaptic terminals, and each presynaptic terminal will discharge the chemicals, and which is sufficient to change the neural potential from minus 65 millivolts to 45 millivolts. So this yes, EPSP produced by the action of excitatory neurotransmitter that depolarizes the postsynaptic memory, and as a result, uh, it opens the sodium or calcium channels, and depolarization of that area happens especially under the presynaptic membrane, and that carries the information. So ESPs are always graded response, large enough to reach the firing level of the action potential is produced, and the postsynaptic potential, what you can see is plus 10 to plus 20 millivolt is needed to produce the action potential. So uh, for the day I will stop here, and I know the synaptic transmission is very complex, and just now I have given you the information, and again probably in the next class again I will uh, explain you again in brief and then move on. So ಏನು ಎಲ್ಲರೂ ಕೇಳ್ತಾ ಇತ್ತಾ?
Yes,
Hello.
sir. Yes, sir.
Okay, then